Hello everyone, welcome to my channel Anupma Biology Classes. In this video, our topic is gibberellins and cytokinins. These are the phytohormones. This video is taken from Plant Physiology Series, CBSE Class 11th Topics, Part 4 of Chapter 16. Now, let's see here the lecture overview. So in this video, we will focus on some important topics related to gibberellins and cytokinins, and which are what are gibberellins, discovery of gibberellins, physiological effects of gibberellins, bioassay of gibberellins, cytokinins, discovery of cytokinins, Zeatin, physiological effects of cytokinins. Now let's start the topic. Here our first topic is gibberellins. This is the second phytohormones. Before this, we discussed about in the previous video, that is auxins, which is the first hormones or plant hormones. So what are gibberellins? The gibberellins are the plant growth hormones which enhance the longitudinal growth of stem when applied to intact plants. However, the gibberellins do not enhance the growth of isolated plant parts. The presence of meristematic cells is necessary for the elongation growth caused by gibberellins. Now, discovery of gibberellins. So, how can gibberellins discovered? Now see here, gibberellins discovered by Kurosawa, a Japanese plant pathologist in 1928. Rice plants infected by the fungus Gibberella fusikuroi showed excessive stem elongation. Symptoms is called Bacne disease. Chemical was extracted and purified and named as gibberellic acid. Now, 80 different gibberellins are available, GA1 to GA80. The most commonly occurring gibberellins is GA3. So, till now about 100 different gibberellins, out of which 25 has been isolated from the fungus Gibberella fusikuroi and 51 from the different higher plants. About 14 are common in both. Gibberellins have been reported to occur in various plant organs such as roots, stem, leaves, buds, flower, roots, nodules, fruits, immature seeds and callus tissues of the higher plants. Gibberellins-like activity has also been reported in the extracts of mosses, algae, ferns, and in the prothallai of Lycopodium japonicum. So, gibberellins plant hormone mainly stimulated elongation of internodes, discovered by Yabuta and Hayashi in 1939, first isolated from the fungus Gibberella fusikuroi. The movement of gibberellins is nonpolar. About 125 types of gibberellins are present. GA1 to GA20 are widely distributed in higher plants. Now, let's see here the physiological effects of gibberellins. So, in case of physiological effects of gibberellins, first is the elongation of stem, then reversal of dwarfism, then bolting on flowering, substitution, the cold treatment, cell division in the cambium, parthenocarpy, seed germination, breaking of dormancy, de novo synthesis of hydrolytic enzymes, and other effects. So the first one, that is here the elongation of stem. Gibberellins produce extraordinary elongation of stems and leaf seeds in intact plants. Many seedlings, when grow in petri dishes containing gibberellic acid 3 solution, show marked elongation of hypocotyl. The elongation of stem is caused due to cell division and cell elongation induced by gibberellic acid. In case of reversal of dwarfism, one of the most striking effects of the gibberellin is the reversal of dwarfism in many genetically dwarf plants. Rosette plant of the sugar beet, when treated with gibberellic acid 3, undergoes marked longitudinal growth of axis. Similarly, dwarf pea plant and dwarf corn attain almost normal size when treated with gibberellins. Extensive studies indicated that the dwarf plants have very low gibberellic acid level, which appear to account for the dwarfing habit. Thus, the external supply of gibberellic acid to such plants causes rapid elongation growth. Next is bolting and flowering. Certain plants show profuse leaf development but reduced, that is retarded internode growth. Such form of growth is called rosette, where a large number of broad leaves remain attached to a very short axis. It has been observed that such plants exhibit excessive internode growth just before reproduction. The axis elongates 5 to 6 times the original height of plant and start flowering. Stimulation in the internode growth just before reproduction is called bolting. Usually, such plants require specific long day period, photoperiods or specific cold requirements to bolt and flower. If such plants are treated with gibberellins during conditions of rosette growth, the plant blot and flower. Next is substituting the cold treatment. Many biennials usually produce flowers in the second year of growth after passing through a winter season. Such plants can substitute the requirement of cold treatment and made to flower in 1 year by the treatment of gibberellic acids. Now, next is cell division in the cambium. So, it has been demonstrated that gibberellins can stimulate cell division in the vascular cambium of many deciduous trees in spring season. After that, parthenocarpy. Formation of seedless fruits without fertilization can also be induced by gibberellin treatment in many plants. These are examples where auxins were ineffective and gibberellin proved to be active in causing parthenocarpy. For example, in case of pome and stone fruits and watermelon also. In case of seed germination, some of the light sensitive seeds Lactuca sativa, barley, etc., can germinate with the treatment of gibberellic acid even in complete dark, which otherwise require specific light conditions. Next is breaking of dormancy. Gibberellins have also been shown to break the dormancy in potato tubers. The dormancy of tree buds during winter can also be broken by exogenous supply at gibberellins. Next is de novo synthesis of hydrolytic enzymes. Gibberellic acid induces de novo a new synthesis of various hydrolytic enzymes such as alpha amylase, ribonuclease, alpha 1,3 glucanase, and proteases in allurone cells of barley grains. This has been experimentally shown by isolating the allurone layer and treating it with the gibberellin solution. These enzymes are mobilized to endosperm during germination and hydrolyze the storage reserve. After this, some other effects like promotion of flowering in the long day plants even under short day conditions, formations of the male flowers on genetically female plants, replacement of female flowers with the male flowers on monoecious plants, and stimulation of pollen germination and growth of pollen tubes in some species are the other effects of the gibberellins. Now, let's see here the bioassay of gibberellins. Seeds of lettuce are germinated in dark at 25 plus minus 1 degree centigrade on wet filter paper. A brief exposure of red light is given during seed germination. Germinated seeds with about 1 to 2 mm hypocotyl are then transferred to petri dishes containing water and gibberellin solutions. The petri dishes are then placed in light for 3 days. After the set period, the length of the hypocotyls are measured. Seedlings treated with the gibberellin solution show marked elongation of hypocotyl. This can be compared with the water control. Now, this is all about the gibberellins. After this, see here what are the main differences between the responses of auxins and gibberellins. Auxin, a plant hormone that is responsible for the elongation of cells in the shoots regulating the plant growth, while the gibberellin is a plant hormone that stimulates stem elongation, germination and flowering. Auxins found in higher plants, while the gibberellin found in some higher plants and fungi. Auxins are double or single unsaturated structure with a side chain, while the gibberellin consists of a mostly saturated tetracyclic gibbon structure without a side chain. Auxin shows a basipetal transport, while the gibberellin shows both basipetal and acropetal transport. Auxin is responsible for cell division, cellular expansion, cell differentiation, axial elongation, lateral expansion, and isodiametric expansion in plants. while the gibberellin responsible for stem elongation, germination, dormancy, flowering, sex expression, enzyme induction, and leaf and fruit senescence. Auxin can promote the growth of shoot segments, while the gibberellin only promotes the growth of the intact shoot. Auxin has a little effect on the leaf growth, while the gibberellin enhances the leaf growth. Auxin leads to apical dominance, while the gibberellin does not cause this apical dominance. Now, see here the cytokinins. The cytokinins are the plant growth substances which act primarily on the cell division and have little or no effect on extension growth. Now, the discovery of cytokinins. Discovery of cytokinins. Here the cytokinins are the plant growth hormones which are basic in nature, either aminopurine or phenylurea derivatives that promote cytokinesis either alone or conjugation with the auxin. Skoog and co-workers found that callus from internodal segments of tobacco proliferate only when in addition to auxin. They were analyzed to find out the growth promoting chemical. The first cytokinin was discovered from the degraded from autoclaved herring sperm DNA by Miller in 1955. Both the auxins and gibberellins causes elongation of plants, but the plant has to grow in whites too. Growth in elongation mainly results by the cell elongation, whereas the growth in white is associated with the active cell division. A cell division factor was isolated by Miller, Skoog, and their collaborators at Wisconsin University, USA in 1955. They were working on the growth of tobacco pith culture and wanted to grow it indefinitely. They added various growth hormones, nutrients, and other substances into the culture, but fruitful results were not obtained. Incidentally, Miller noticed on an old bottle of DNA kept for several years in the lab. He added the contents of bottle into the culture medium, and to his surprise, the old stock of the nucleic acid could support and hasten the growth of tobacco callus tissue over an extended period. Miller and Skoog obtained similar results with yeast extract. Fresh DNA, when added in the medium, failed to give positive results. Therefore, they formulated the idea that the cell division factor is some degradation product of the DNA. Later on, Miller and his colleagues isolated and purified their substance in crystalline form from herring sperm DNA. This substance was identified as 6-phosphorylaminopurine. The name this compound kinetin because of its property to activate cell division. Here you can see the structure of kinetin. Following the discovery of kinetin, many other compounds were discovered which showed similar activity. Scientists then proved the term kinine for the group of such compounds since the term kinine was already existing in relation to animals. Skoog and others gave the term cytokinin for all those compounds that exhibit kinetin-like activities. This is the discovery of cytokinins. Now, what is Zeatin? Zeatin, it is a cytokinin-like substance isolated and crystallized by lithium from milky endosperm of corn, Gia maize. He named this substance as Zeatin. This substance was found to be first naturally occurring cytokinin in plants. Later on, the substance was characterized as 4-hydroxy-3-methyl-trans-2-butynyl aminopurines. Further studies have shown that Zeatin is also present in the root, stem, and leaves of maize plant, but in lesser amounts as compared to milk stage kernels. Since then, several other cytokinins have been reported in various other plants. Now, the physiological effects of cytokinins. So, like the cell division, cell enlargement, initiation of interfascicular cambium, morphogenesis, counteractions of the apical dominance, breaking of dormancy, delay of senescence, accumulation and translocation of solutes, and the other effects. So, first one is the cell division. The most important function of cytokinin is the promotion of cell division. It is now well established that these are the true cell division factors. The varying amount of cytokinins along with the sufficient auxin is required for the growth of callus is tissue culture experiment. In case of cell enlargement, the cytokinins can also cause the enlargement of cell in leaf discs and cotyledons. This effect of kinetin can occur in the absence of auxin. Kinetin also causes cell enlargements in tobacco pith culture and tobacco roots. The cell enlargement due to kinetin is partly due to stimulated water uptake and partly due to increased plasticity of the cell walls. Now, the next is initiation of interfascicular cambium. The cytokinins can induce the formation of interfascicular cambium in plants. After this, morphogenesis. Cytokinins play a vital role in the morphogenesis in plants. It opposes initiation of roots in stem cutting treated with auxins and results in the formation of callus at the cutting. It is now well known that the kinetin-auxin interactions control the morphogenetic differentiation of shoot and root meristems. Now, counteraction of apical dominance. External application of cytokinins promote the growth of lateral buds even if the apical bud is intact. Thus, the cytokinins reverse the auxin-induced inhibition of lateral buds and counteract the apical dominance. Next is breaking of dormancy. Cytokinins can break the dormancy of many seeds and also promote their germination. After this, delay of senescence. Senescence is the phenomena in which the mature leaves lose their pigment chlorophyll, turn yellow proteins and degraded, and ultimately they shed from the plants. Richmond and Lang, while working on detached xanthium leaves, observed that application of cytokinin delays the process of senescence for a number of days. The phenomena of delaying senescence by cytokinin is known as Richmond-Lang effect, and this physiological effect of cytokinin has now been observed in many plants and helps them to remain green much longer by reducing the breakdown of chlorophyll, proteins and nucleic acids. Next is the accumulation and translocation of solutes. Plants accumulate solutes very actively with the help of cytokinins. A high production of cytokinin in some plants help them to grow stunted. So, cytokinins also help solute translocation in phloem. During development, it helps phloem to carry more nitrogen and other nutrients to the site where they are required. Some other effects like the induction of plastid differentiation, differentiation of interfascicular cambium and lignification. Cytokinins provide resistance to plant under high temperature, replacement of photoperiodic requirement for flowering in the certain plants, stimulation of several enzymes, especially those concerned with the photosynthesis, etc. So these are the physiological effects of cytokinins. Now, this is the end of video in which we knew about the gibberellins and cytokinins in detail. The next upcoming video will be on the ethylene and abscisic acid. So if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any question, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.